Hi, good afternoon, everyone. So, welcome to our another weekly Terasaki talk. Today, our presenter is Professor Danielle Benoit. So, her topic is innate immunity, front and foe in the development of therapeutic biomaterials. Maybe you have seen Muhammad always in front of this one. Today is my turn, and this is my first time I'm doing. Dr. Benoit, if I want to talk about her, there are plenty of things we can talk. Dr. Benoit is the inaugural Lorry Lockey Chair and Professor in the Department of Bioengineering at the University of Oregon Knight Campus. Professor Benoit directs an internationally recognized research program focused on the therapeutic biomaterial with an emphasis on challenging clinical problems and a translational solution involving cell transplantation and drug delivery. Actually, during my postdoc time also, I was closely watching her research, especially the periosteum engineering. I was very well impressed with her research, and I cited a couple of my articles also, her work. Actually, it is very interesting. If you are interested to see more about the periosteum engineering, she's the best person to look up. If you can, you will get more information from her. And moreover, she got plenty of awards. For example, Professor Benoit has been recognized by numerous awards and for her research, including 2019 class of AIMB Fellows, 2018 University of Maine Distinguished Alumni Award, in 2016 Clayton Gleason Young Engineer of the Year Award, 2015 Young Innovator Award in BMES, and NSF Career Award also she got in. And she served different NIH study section, especially she was the standing member in the NIH BMI study section. So thank you, Dr. Benoit, for accepting the in-person visit. And we are excited about your talk. Welcome.
Wonderful. Thank you so much, Johnson. And actually, I have to admit that I got invited to give a Terasaki talk. And I was like, can I come in person? So it's actually, thank you for letting me come and present in person and learn more about the really amazing and innovative work that you're doing here. So thank you. And thank you, Johnson, very much. So apologies. I think that my initial talk title was different from this, and we just realized this just now. I was like, oh, I could bring that up. It could be cool. It's fine. But Johnson was very nice and said, go ahead and use this talk. So thank you. And I do want to say that, again, I've had a wonderful visit, and thank you for your graciousness, and really excited to share some of our work. So let's see if we're going to get this to work, right? It's going to move. There we go. Okay, now it's going to skip ahead like 16 slides. So my work is in therapeutic biomaterials. So we work with polymeric systems. And so we use a couple different flavors of biomaterials in my group. And I will predominantly talk about the polymers and the polymeric systems on the left, that we use a controlled living polymerization known as reversible addition fragmentation chain for polymerization to make these. So that's a mouthful. So we shorten that to RAFT. So RAFT polymerization has been around for a couple of decades, and we only scratched the surface in terms of its capabilities. What we really like about RAFT is that you can take a typical radical mediated polymerization process and just introduce a chain transfer agent, and if it's properly designed to mesh well with the monomers functionalities that you're using, and I'm just showing one example of a dithiocarbamate with a generic RNZ group, you can form polymers with high control. So what does that mean? That means that the molecular weight and polydispersity can be successfully generated from day to day if you have the right controlled experiments and the right controlled polymerization approach. What's also really great about this approach is that it's living. So not only do you have very good reproducibility of your polymers day to day, but you can stop the polymerization and restart it in the presence of another monomer. So you can make polymer red and stop the reaction and introduce monomer blue and make a dye block of a red block and then a blue block. And of course the world is your oyster with regards to the design parameters that you can explore here. So it doesn't have to be a red monomer and a blue monomer. You could have the whole rainbow of monomers. So, those monomers have different functionalities. And so it gives us a really neat tool to develop some really sophisticated and controlled delivery strategies. And so hopefully I will convince you that this is useful as a mechanism through my talk. So I will predominantly talk about dye blocks today. Again, I said we scratched the surface in terms of the capabilities of Raft. We've dabbled in using brush copolymers, but there's a whole world out there in terms of capabilities. So it's a really exciting opportunity for anybody who's exploring new drug delivery systems. Where I'm not going to talk much about, or really at all besides this slide today, is our use of hydrogel materials. So in situ forming photopolymers in particular. So we still use, so this is polyethylene glycol. Hopefully some of you recognize this as a core. And I'm showing a methacrylated or dimethacrylated polyethylene glycol. We don't use this a lot in my lab, but it's an easy way to show this chemistry. In the presence of initiator and light, these methacrylate groups can react together to form polymethyl methacrylate that is then cross-linked through this polyethylene glycol. And because PEG is so hydrophilic, and you can change the molecular weight and also the functionality of PEG, because it loves water so much, you end up with this hydrogel. So much like Jello is what I always give the visualization as. So it's just like Jello, and it's akin, from a very broad description, akin to your native extracellular matrix, that it's mostly water. Where it's not is it lacks all the functionality of your extracellular matrix body. So it gives us the ability to tune and introduce biofunctionalization, degradation, and mediated through enzymatic activity and otherwise. So we use this in tissue engineering approaches. We use this, and here I'm showing just encapsulated mesenchymal stem cells that are mostly living after being trapped. We also have delved into, as many of you have, in developing tissues on a chip and starting to develop microphysiological models, leveraging this type of material platform. Okay, so within this realm, we have a whole host of different projects within my group, and the slides will advance, I promise. I think. Here's hoping it's going to happen. Maybe it's because I'm using the laser pointer too. Oh, there we go. Okay, so this is a horrible slide. I apologize. I keep saying to myself that if ever my student presented this, I would chastise them very much so, because this isn't just something you should never present. But it is a very useful tool to show that we work in, again, polymeric drug delivery strategies and then hydrogels, and the variety of applications that we delve into. And I'm gonna focus on a couple of these over here, and we'll get to those in the next slide. But in terms of the hydrogel work, as Johnson already alluded to, we engineer the periosteum and try to develop strategies to improve bone healing in that way. We use our hydrogel depots to deliver in a controlled way a variety of drugs, including peptides, and siRNA nanoparticles, which I'll tell you a little bit more today, and then developing microphysiological systems for the salivary gland and also for retinal tissue. And I'm happy to discuss this more if you have questions. So where I'm going to start today is RNAi, or RNA interference methodologies. And I don't think that this really needs an introduction, but I think most of us are well aware of the huge potential and therapeutic, even clinical success of RNA interference technology and RNA delivery, and how it's impacted our daily lives, including the COVID vaccine, the booster that I got on Sunday, which is still hurting my arm, by the way. So siRNA is the type of molecule that I've been very exclusively interested in some of our work. So it modulates post-transcriptional gene silencing in a mechanism that's shown here on the right. So siRNA can be introduced into the cell cytoplasm, and similar to endogenous microRNAs, can be introduced to the RNA-induced silencing complex and then modulate through Watson-Crick base pairing hybridization with mRNA, which then reduces or blocks translation. It can also actually cause the catalytic destruction of mRNA, which is, and I say it's catalytic, that activated RISC complex can continue to degrade mRNA over and over. So what's beautiful about this is that you can silence, in theory, any gene with a known sequence. It becomes a little bit tricky once you introduce things like protein half-life, but that's something that we've learned over the years and others have as well. But it's a really beautiful technique. So this is old data. So in 2015, there was 30 clinical trials. So today, there's actually still only about 30 clinical trials, but of course, the ones in 2015 have moved through the clinic, and some have been successes, right? So we have 5 nucleic acid or siRNA-based therapeutics on the market now. So not the 2 that I've listed here. The newest data is there's now 5. What's interesting about this is that the 5 that are available are all based on liver therapeutics. So based on liver diseases, treating things in the liver. And this is because when we introduce nanoparticles to the body, as all of you or know, things go to the liver pretty readily. And so, delivery barriers are really a critical hurdle still in this nucleic acid delivery. And, it involves, systemic circulation. We'll talk about that a little bit today in our work. But also intracellular cellular barriers like low cellular uptake and degradation, like endolysosomally. So lack of delivery systems is an issue. We've spent a lot of time developing a drug delivery paradigm, so a drug delivery system that enables tuning actually different cell types. So we've published widely in this. I'm not going to cover it too much, but I will tell you a little bit about how we designed in the different functionalities of this diblock copolymer, which is made of dimethylaminoethyl methacrylate blocked with a tercopolymer block of dimethylaminoethyl methacrylate, propylacrylic acid, and butyl-methacrylate. And just owing to the hydrophobicity of the second block, these become nanoparticles of about 25 or 30 nanometers in the body that are cationic and just ready to electrostatically bind with nucleic acids. So this is very specially designed. So we've really been careful in terms of our design parameters in terms of this diblock copolymer, where the first block is specifically designed for siRNA complexation. So dimethylaminoethyl methacrylate, this first block, is partially protonated 50% at physiological conditions. So this gives us cationic characteristics, which enables these nanoparticles to then bind with siRNA. As shown here, so complexation, and that actually modulates uptake. So even though we're complexing, we're actually maintaining a positive overall surface charge of these nanoparticles, which enable co-localization electrostatically with the cell membrane and introduction into the cell through endosomal processes. The second block is also very specially designed. So again, we have a partially protonated state in our dimethylaminoethyl methacrylate. Likewise, we have a partially negative charge here in the propylacrylic acid. So overall, at physiological conditions, the second block is neutral, right? So it's partially protonated, partially negatively charged, so amphilytic. But what happens when we enter the endosome, the pH drops. And so what happens is that the dimethylaminoethyl methacrylate becomes fully protonated, and as does the propylacrylic acid. And all of a sudden you have this electrostatic repulsion now due to charges in that core block, which then modulate disruption of this nanoparticle and then interaction with the cellular or endosomal membrane, and then release of siRNA into the cell cytoplasm. So this is how we're actually able to then interact with the RNA-induced silencing complex. So again, very, very specially designed in terms of the overall design parameters for this block, or diblock copolymer. So when I started my lab, so going on 13 years ago, I had a dream that we could use siRNA delivery to control mesenchymal stem cell fate. So I had lots of interest in regenerative medicine, and there were it's very complex, like how you get even a mesenchymal stem cell or marrow stromal cell, whatever term you use these days, how you get those cells to differentiate into specific cell types. There's growth factors there. Now there's a whole host of new things too, like stiffness, material stiffness, but there was growth factors, there was small molecule drugs, there was now stiffness and other things. So all of these, though, become integrated intracellularly in the cells to then execute on those differentiation pathways. So there was a paradigm, and I think it still exists, that, well, what if we can, instead of giving all these external stimuli, we give intracellular stimuli to then bypass all of those complex cues and initiate differentiation intracellularly through microRNA like siRNAs, acting through those microRNA pathways. So this is my grandiose dream, and I'm going to tell you that it was a complicated path. So anyway, that's where we started. We were very excited about this opportunity, and MSCs have self-renewal. They can be isolated and expanded for patient-specific therapies. They secrete trophic factors, and they have this wonderful multi-lineage potential. And again, my dream, and I think that it's still possible, it's just more complicated than this, as most things are, that you can control that via siRNA delivery if you pick the right ones. So starting down this path, one of my graduate students said, okay, well, let's design these diblock copolymers to be very selective to and modified and optimized for MSCs, because the generic diblock copolymer, I explained how it works, but certainly the endosomal lysosomal pathway, the uptake mechanisms, all of that is different depending on which cell type you're using, right? And so we kind of went along this path to identify, well, if we have MSCs versus actually, human versus mouse, or MSCs versus tenocytes or other cells that we were interested in at the time, how could we optimize the parameters of delivery? So we did things like optimizing on the first block, so the corona block, molecular weight. Second block, the corona to core, and the amount of butylmethacrylate in that second block. So these are things that we could change. The diameter generally stayed similar across these different diblock copolymers and self-assembled nanoparticles. And the surface charge also was positive, suggesting colloidal stability. But when we did all of these different parameterization and design of experiments to identify the optimum parameters, we found our optimum had a large amount of butylmethacrylate and a large first block to second block ratio. So I'm just showing you the data here to just kind of skip over some of the details. Happy to share them with you later. But what we were able to find is that when we looked at untreated versus siRNA alone versus a positive control, our nanoparticle was able to execute on siRNA delivery. So we had high siRNA positive MSCs, and the mean fluorescent intensity was just kind of an averaging over the amount of siRNA that's been internalized of these cells, was also quite good. We could achieve good gene silencing. So this is just a housekeeping gene, that one of many that we use, PPIB, and we got good knockdown. And very good survivability. So viability at 24 hours was quite good. So this is all great. Like, this is a whole study, right? So when it got interesting was when we started to take those nanoparticles that we had optimized for the MSC siRNA delivery and start to deliver siRNAs that were implicated in osteogenesis in particular. So we did this for a few, but I'll show you the osteogenic ones. These are easier, makes for an easier story, just because we can use morphology. So of course we ran controls, right? Controls are very important. They were very important for this study. We introduced our MSCs in the context of osteogenic media. This is like the typical cocktail that you'll see in the literature. So this is dexamethasone, beta-ascorbic acid, and beta-glycerophosphate. So very typical, very similar what many people do in the field. And sure enough, after 7 days, you can see that these cells are starting to form into more, osteogenic-like phenotype based on their morphology. We did a bunch of other characterization, but the morphology is pretty interesting and easy to show. So they're starting to form these cuboidal-like structures that's consistent with osteoblasts. When we looked at, so this is non-targeted siRNA delivered with our nanoparticles, we saw a dramatically different phenotype. So this is a control where we are treating with osteogenic media, but our morphology, and again, gene expression and otherwise, also bore the same conclusion that these cells aren't differentiating anymore. So something is happening in terms of the treatment, and it's not anything to do with the siRNA, because this is, again, a non-targeted siRNA, or it's not, it's counter to our overall thesis that, if we can deliver then an osteogenic siRNA in the absence of osteogenic media, we should be able to induce differentiation. But this is interesting enough, and we repeated it a number of times, that we had to follow up, right? So something's happening that's inhibiting differentiation. So we did a bunch of experiments and finally threw our hands in the air and said, okay, let's do RNA-seq to identify what might be happening with these cells. So what we found was there was an enrichment in innate immune cell signaling. So, and this is just looking at nanoparticle or non-treated cells versus the nanoparticle siRNA, and this is non-targeted siRNA at days 1, 5, and 14. And we did a bunch of, might be able to squint and see some stuff in the background. There's a bunch of other groups that we ran, but this is where the rubber hits the road. So I'm just going to tell you a little bit about this. We saw NF-kappa B signaling up. We saw toll-like receptor signaling up. We saw RIG-like receptor signaling up. All are implicated in inhibiting differentiation and are all, intracellular, just innate immune signaling pathways. So, okay, so it's a thing. So now we know what's happening intracellularly, but why? Why is this happening? So we also explored this in a variety of different ways. We used locked nucleic acids to kind of knock out the toll receptor signal. That just won the Nobel Prize for people that had figured out the way to modify nucleic acids to prevent that. That didn't make a difference. It's a little bit more difficult to explore NF-kappa B and retinoic acid. So we did a variety of other things looking at metabolic activity and otherwise, and still we're just kind of stuck. So it wasn't until we looked just using EM and looked at the cells. So, very globally looking at the morphology of different organelles. And what we found is that, no obvious differences in mitochondria, no real changes in ER. But what did strike us is when we looked at the membrane. And what we found was that there were nanoparticles that were sitting right at the membrane that were much larger than they should have been in terms of the nanoparticles, which were only about 25 nanometers. So that caused us to explore this a little bit more. So for those of you working in nanoparticles or nanotechnology, you're probably well aware of the challenges of analyzing nanoparticle size when you have serum, for example, at the typical levels that we use, because what gets washed out is your nanoparticles, and all you're measuring is BSA size in using, dynamic light scattering, for example. So we hadn't very rigorously looked at this. I had a really talented graduate student at the time who was determined to be able to measure this. So he did some really phenomenal things in terms of incubating the nanoparticles with serum and then reducing the concentration and being able, using dialysis to be able to look at this. And sure enough, what his stubbornness did pay off, or determination, I should say, paid off. And what we showed is that in the presence of any amount of serum that's 2% or higher, we got this huge increase in size of our nanoparticles. So these particles are becoming glued together by the serum that we're using in our cell culture. And that's also borne out from the polydispersity index. And he also had to jump through some significant hoops to get electron microscopy, because you talk to any electron microscopist and they're like, do not put protein in my expensive microscope. Again, protein is like glue. Glue is not good in an electron microscope. So we ended up using actually hydrogels to entrap these particles to allow for imaging so we could wash away the protein as best as we can. But sure enough, in the absence of protein, you see nice unimeric nanoparticles and big giant blobs in the presence of protein. So serum, okay. So we also explored the efficacy. So amongst other things, so we started to use no serum conditions, and sure enough, even at 10 nanomolar siRNA concentrations, we could get almost 97% or better knockdown of PPIB, which is really dramatically different from the 30 nanomolar with 10% FBS. So okay, all right, so we're learning. So I said to my student at the time, okay, so it seems like we're, at an impasse, but talk to me at the same time. I had lots of parallel experiments going on in my group that had worked, in terms of siRNA delivery, okay? So for example, we had retroductally delivered siRNA to the salivary gland. Yes, the salivary gland, you heard that right. So we have a very interesting and long-standing collaboration now on trying to identify methods to prevent off-target radiation damage to the salivary gland, and one of the approaches that we took early on in my group, again, in a collaborative effort. And what we showed was that indeed we could deliver an siRNA that should prevent radiation, and we did. We maintained saliva secretion because that's the big product from our salivary glands is saliva. So this worked, asterisks. We had delivered siRNA nanoparticles locally to fractures and inhibited an inhibitor fracture healing, WWP1, and showed that we could rescue or rescue the rate of fracture healing quite significantly compared to no treatment or some other controls that I'm showing here. So this worked, asterisks. We had also locally delivered siRNA nanoparticles to the tendon, and with beyond the nanoparticles, just the delivery as the delivery mechanism, and we could improve stress, ultimate stress, at a 4-week time point compared to scrambled control. So I said to my student Dominic, so why did these work? Asterisks. And you're showing me that they shouldn't because of this protein adsorption characteristic and gluing together of the nanoparticles. So we kind of thought about this a little bit and came up with a really key experiment that highlighted why these could work, and now no asterisks. And the reason is because of the concentration of protein that exists in these local sites. So these are, retroductally injected to the gland. They are locally injected to the tendon or locally released in the bone fracture site. And the concentration of proteins in those that extracellular matrix is far lower than it is in any serum that you would use for cell culture. So serum albumin is 40, and I'm just showing albumin as one of the many components, but it's a very large component. 40 grams per liter. Interstitial fluid, much lower, anywhere from 10 to 20, depending upon what fluid you're looking at in the body. Salivary albumin, even smaller. So to kind of really close the loop on this, we then did more retroductal injections and asked, are these particles, are they glommed together, are they aggregated, or are they unimeric that would be useful? And what we found is that indeed they're singular, and you can look at that a little bit closer here and this is within the ductal epithelium. So this is quite intense in terms of being able to find this in the ductal epithelium. But sure enough, we could find these nanoparticles that were separated, so fully unimeric and able to deliver their cargo most effectively. So to now, I've shown you that protein is a challenging thing in terms of aggregating nanoparticles. So how do we combat that? And I guess maybe more importantly, this aggregated nanoparticle sets off a whole host of cascade within cells that can impact very much their differentiation potential. So then we asked, can we reduce protein absorption. I mean, this has been something in the biomaterials field since it started, right? Anti-fouling materials. So, you know, when my next student, different student, was so grandiose in saying, I think we can make fouling surfaces. And I'm like, well, we'll see. Maybe we can make selective protein binding surfaces, but we've actually made a pretty good pass at this. So what I've shown you is that serum proteins bind, and this causes a lot of downstream effects, loss of function. It impacts delivery and downstream, effects of drugs that we're delivering, including siRNA. And so this aggregation too, you can see it very clearly in terms of electron microscopy. So how do you reduce that, or at least modulate it or control it? And of course, you're all thinking, well, what? You already talked about PEG. You must be aware of its properties, and absolutely we are. And we're very, very lucky that polyethylene glycol exists and has been used to sustain circulation time in a variety of nanotherapeutics, and it works wonders. There's some issues, I think, with some recognition these days that may or may not cause it to be discontinued or maybe reduction in use. There's also carboxybetanes that have similar anti-fouling behavior through zwitterionic behavior. So really protein-loving type of molecules like carboxybetane. Along a similar line, there are peptides that exist that have amphilytic behavior, so both cationic and anionic behavior that have been used, and really leveraging this water-loving activity. But what Clyde really wanted to do was boil this down to thermodynamics. So the reason that protein doesn't bind to PEG or doesn't bind to carboxybetane or doesn't bind to some of these amphilytic peptides is because water is, these are water-loving entities, and so you have to push the water out of the way in order to get protein to absorb. So really, that's a thermodynamically driven process. Where with low protein absorption energy, you're going to maintain the numeric structure. But with high protein absorption, you're going to cause aggregation. So can we address this and design surface modifications that would just use this as a parameter, just thermodynamics, boil it down to thermodynamics, to have a little bit more agnostic and greater design space to enable this, reduction in protein absorption. So, that's exactly what he did. And he decided to use peptides because there's a nice range of functionalities and many different native amino acids, but then also non-native amino acids that one could use. So you can introduce positively charged amino acids and negatively charged amino acids, again, to get this amphilytic behavior, zwitterionic behavior. And then also using neutral amino acids, which kind of helps space the peptides out to keep them, in that water-loving state as opposed to protein-loving state. And so he boiled it down to charged sequences even. He didn't even specify lysine or arginine or histidine or any of these charged sequences. He just boiled it down to positive, negative, neutral. And then ask questions about, okay, how are they going to bind to proteins? And what is that delta G, right? The Gibbs free energy of binding. So he could generate peptides and then actually use a computer algorithm that had been developed called PASTA. So this is from an Italian group. So they developed this computer algorithm called PASTA, and I don't remember what this acronym stands for, but it's actually to predict aggregation behavior of proteins. So you could flip that on its head and say, how do you anti-aggregate proteins? And that's what Clyde leveraged. But it does boil down to this delta G parameter. So then, from the hits, and actually he selected for those that had low energy and those that had a high energy, and kind of binned hits and said, I'm going to make, a certain number, and there's only you got to keep this feasible. So, only a select number, but a good number in each bin. and then introduce these to the nanoparticles. And of course, I'm skipping over a whole bunch of work, right? So sorry, Clyde. But what he found was that, he could bend these, and then the predicted activity would be that the nanostructures would stay intact and not aggregate, or they would with the high energy. And that's exactly what he found. So introduced serum proteins. So this is 10% serum. And what he found with the low energy zips is that he could reduce protein absorption substantially through these low energy zwitterionic peptides, which we're shortening to zips. It's actually, in combination or collation of all of these low energy zips can outperform PEG in protein absorption. The high energy actually increases protein absorption, which we were really concerned that we couldn't possibly do that because these aggregated so terribly, but we were able to do that. We then looked at diameter measures, and indeed we could reduce protein, the size of the nanoparticles, similar to PEG, and the high energy, of course, is still aggregated. And then we looked at delivery, and again, of siRNA in this case against GAPDH. So I'm switching up your housekeeping gene, but the behavior is similar. So here's nanoparticle alone with low energy, and PEG. PEG outperforms our low energy zips when we sum over all of their activity. But still, it does a fairly good job to enhance siRNA delivery and efficacy. So that's pretty exciting. The other thing we wanted to test was for circulation time. So this should also be improved with the low energy zips. And so this is just imaging an ear of a mouse after injection of the nanoparticles that are fluorescent. And what we can show is quantitatively we can improve circulation time by about 2x. I think that the fold improvement, this is on the order of PEG. We don't have that control here directly linked, but I think that we're close to the PEGylated system in terms of the improvement in circulation time. Okay, so yes, I have time. So what I've been able to show you is that we've developed a diblock copolymer, what I would call a family of polymers, and really optimized parameters to improve delivery to a variety of cell types, actually, not just MSCs. But what we found is that aggregation is a real problem, and it induces a lot of innate immunity processes within cells, which was something that I had to learn a lot about once we discovered this. And we can prevent that by blocking protein absorption and aggregation by introducing zwitterionic peptides that are specially designed to reduce protein absorption. What's cool about the zips too that I didn't highlight today, but they can actually preferentially allow for absorption of different proteins. So we're starting to explore that too. So it's a pretty interesting phenomenon, and a nice new winding path for us to follow. Okay. So I'm super excited that I have time to talk about the next part. So hopefully you gathered that was an immunity foe, but how we can kind of like co-opt the innate immunity nevertheless. So the next segment, and this will be it, I'm not going to try to jam another story in, just two, is in our development of bone-targeted drug delivery systems. So this is another long-standing project that has waxed and waned and taken deep dives in places I never thought I would be. But hopefully I'm going to be able to show you that innate immune system can actually be our friend. So I don't need to belabor it here in this audience, but drug delivery is a real challenge. So traditional drugs have rapid clearance mechanisms, poor biodistribution, especially to bone, so less than 1% gets to bone. And toxicity, therefore, because you're trying to hit the sweet spot between therapeutic efficacy and toxicity. And so as you have poor biodistribution, rapid clearance, you can jack up concentrations that you're delivering, but then you end up reaching into that toxicity realm. So it's a real challenge. And so we've addressed this, of course, using a drug delivery system. So this is another one that we use RAF polymerization to synthesize. And this is a really, really neat diblock copolymer that we've, again, leveraged for a variety of uses. But what's beautiful about this is that it's a one-pot reaction. So I sold Raft that you can make, sprinkle in your one monomer and stop the reaction, then sprinkle in your new monomer and have your second block synthesized. So what's great about this particular polymer is that styrene and maleic anhydride will preferentially react with each other until you exhaust the maleic anhydride. And at which point, when you have an excess of styrene, the styrene will just pick up and you'll form your diblock in one pot in one reaction. So makes for a very easy synthesis. So ultimately, we achieved this diblock copolymer shown here that is an alternating diblock of styrene and maleic anhydride. And of course, maleic anhydride will readily ring open in the presence of water, offering you 2 carboxylic acids that make this a very hydrophilic and negatively charged block. And then a styrene core, which gives you nice crystalline behavior within the core. So this diblock, I'm going to simplify. So the chemistry that's on the ends, of course, is the chain transfer agent, and it's just not super important for our discussion today, but very useful. So happy to tell you a little bit more about how useful it is, but not super important for today. So we have this hydrophilic first block and hydrophobic second block. So just like the first discussion with our diblock copolymer, this also will readily self-assemble in water, forming about 25 nanometer particles. And under electron microscopy, they do actually kind of look like the cartoon that I'm displaying here. They're hairy little beasts. In a scintillation vial, they look like this. So like a little bit milky water, blue hued based on the size. And again, we've used these quite extensively. And I'll tell you about snippets of these different papers and what we've done with these. To target bone, so to develop targeted drug delivery systems for bone. And there's a variety of ways to target tissues that have been used across the literature. And in our system, we use a peptide, and this is because of some really formative work about, actually, this work was motivated to understand how bone resorption and production is coupled. But what came out of this is that when bone is being resorbed by osteoclasts, they leave behind a marker that we know well in the bone field known as tartrate-resistant acid phosphatase. So this is just a protein that the osteoclasts deposit as they're chewing up the bone and getting ready to be remodeled. So some work that predates me, actually, but by a collaborator of mine, they had developed these bone pits in cortical bone wafers, so through activating osteoclasts and having them eat up and develop these excavation pits. And here's the trap that I've drawn onto that because you can't possibly see it. And then they use phage display to identify what kind of peptides might be being used that are parts of proteins on osteoblasts to recognize where we need to deposit bone. So that was a whole motivation for this study, right? So they use the phage display, they use a very directed cDNA library on the osteoblasts, and then identified, well, what peptides are then binding to trap, and then we're able to then come back out and say, this peptide is in these different membrane proteins of osteoblasts. So did phage display and identified what we now call trap binding peptide, tartrate-resistant acid phosphatase binding peptide. Has super high affinity in terms of the phage. So this is the phage affinity. So it's a little bit unfair because there's several displays of this peptide. When my collaborator and I had this discussion in 2009, I think it was, we were chatting and he's like, so is this interesting? And I was like, yes, Ed, this is really cool. I think that this would be a really handy way to target drugs to bone. Well, didn't that take a long time to suss out, but I'm happy to say that it works. But there's a good bit in between here and there that I'll fill in some of the gaps. So, and of course I'm simplifying this. This has been, painstaking process for a variety of people in my group, as it always is. But what we've been able to do is introduce this tartrate-resistant acid phosphatase peptide, which is shown here in the simple one-letter amino acid sequence. And you'll see this alloc group that protects the lysine, just so we can do this selectively. So we can introduce via the primary amine and not the amines on the lysine into our nanoparticles. And we actually do this ring opening that maleic anhydride. So we can ring open the maleic anhydride to introduce this peptide and then deprotect that alloc. And ask questions, does this work? Well, I already told you that it did, but let's give you some evidence. So we can introduce the nanoparticles into a fracture model. So we use a femur fracture model in mice. Nanoparticles already accumulate on their own to the fracture site, and that's well known through what at least we like to call is enhanced permeability and retention. So this is because there's leaky vasculature within the fracture site. So nanoparticles accumulate. The scrambled peptide that we use also accumulates similar to the nanoparticles alone. And then the targeted actually does have greater accumulation in terms of overall intensity. We can quantitate that. So we can quantitate this and we see, exactly what the images showed or suggested, that we get an enhancement in accumulation through the targeted approach. And I've lost my significance bars, but I promise that these are significant versus these controls and versus saline. We can look at the fracture too. And other tissues. So, not surprisingly, we see a significant amount of nanoparticles in liver and spleen. Where it gets really interesting though, and deviations and shows some deviation quite extensively across the different nanoparticles, is at the fractured bone. And so what you see here with nanoparticle alone, and here's your fracture site right there. So you can kind of squint and kind of see where the fracture was. And very, very strikingly, very little pink and not nanoparticles alone, very little with scrambled peptide functionalized nanoparticles. And it is the targeted peptide nanoparticles that really have that very intense pink color that's consistent with the IR780 that's been loaded as a model drug and also fluorophore for these studies. Okay, great, you can deliver fluorophore. What about something that might be helpful? Oh, sorry, I quantitated it too here, or I shouldn't say I. My really wonderful graduate student, Yu-Chen Wang, quantified the accumulation here. And what's interesting is that as we introduce the targeting group, we see reductions in liver, spleen, and lung, increase in unfractured bone. So it's not surprising. TRAP is not specifically expressed only when there's a fracture. It's also throughout all of bone. But fractured bone definitely has the majority. Okay. So we can target to bone and then release drugs that should have an osteogenic effect, right? So what might they be? So what kind of drugs might we want to use in this scenario? And I promise I'll try to zip through the next 5 minutes, the next bit in 5 minutes or less. So we have some time for discussion. So in the context of differentiation of MSCs to osteoprogenitor cells, and then osteoblasts and chondrocytes, it's really in the non-union sites, which is a deficiency, similar to deficiencies in healing or have deficiencies in healing. It's really this osteogenic pathway that's inhibited. So we targeted specifically or looked for drugs that we could target here in terms of the downstream pathway of osteogenesis. And Wnt came up as a winner because there's many Wnt agonists that have been developed and have been through at least phase 1 and 2 clinical trials. So Wnt is, a signaling pathway that involves many different proteins. So at the cell membrane surface, LRP5 and 6 and frizzled. There's this multi-protein complex that has a GSK3 beta involved in it that mediates ubiquitination of beta-catenin, which causes degradation. So then, therefore, there's not downstream signaling that turns on. But if you can somehow, if you turn on Wnt signaling, that then alleviates that ubiquitination, beta-catenin accumulates and can get turned on, and turn on osteogenic genes like RUNX and COL1. So we can avoid extracellular signaling by introducing a GSK3 beta inhibitor. So blocking activity, blocking the ubiquitination, and we can do that quite effectively to improve beta-catenin levels. So we can introduce this GSK3 beta inhibitor. It loads quite effectively with high loading capacity, and we can deliver it to our fractures. And what's really exciting is that we see is with our targeted drug delivery system versus all of these controls, saline-free drug and scrambled peptide, we see a rapid conversion. This is histology looking at cartilage right at that fracture site and bone and soft tissue. We see a really much faster conversion of the cartilage into the bone and soft tissue only within the targeted drug delivery system, whereas we still see cartilage even at 4 weeks with the other treatments. We can also see expedited healing in the context of micro-CT. So this is just the imagery, but I hope you can see that the bone is almost fully restored in the context of the week 4 targeted drug delivery system. We can quantitate that and again see significant improvements with the targeted drug delivery system versus the other controls. And finally, bone is biomechanical. So you should always test the biomechanics. So we tested torsional rigidity. We have a bunch of other measures, but this is the one that I'm showing here. And we do see an improvement in torsional rigidity at 4 weeks after fracture. So that's super exciting that we can rescue uninjured bone levels in just 4 weeks. So we had this model where we can target drug delivery to what we thought was the osteoblast to improve healing. And that's where things got interesting again, where when we looked at the fractured bone and looked at the positive cells, it is not the osteoblast or even MSCs that are positive for these nanoparticles. It's the macrophage. So that caused us to rethink this model a little bit. So it seems like the healing is actually working through macrophages. And we are well aware now that in the field that macrophages play a really important role in bone healing. So they're recruited readily at the start. They turn into from monocytes into M1 macrophages to start cleaning up the debris. Then they transition to M2 phenotypes and coordinate cells like endothelial cells and a variety of other cell types, and even coordinate osteoblast differentiation through soluble cues, and finally, with the restoration of bone, are still continuing to play a role through their ability to differentiate into osteoclasts. So clearly, macrophages are important, and we are delivering this at this early phase. So right at the end of the inflammatory phase is when our drug delivery system is delivered. And so when we look at the macrophage at fractures, I'm looking just at free drug and the targeted drug in this experiment. And what we find is that when we look for orange staining, which is macrophages that are M2 phenotype, marked by F480 and CD206, there's some here at 10 days post-fracture with just drug alone, but it's very much enriched in the context of our targeted drug delivery system. And moreover, we can see a variety of cells that are also doubly positive here at the cortical bone surface. So here at this red box, and this is suggesting that osteomacs or osteomacrophages are being restored, which also is a marker for enhanced healing and much more rapid healing, the reestablishment of this cell population. So we also wanted to do RNA-seq, and I'm going to really give the high glossy conclusion from this. So what you see here is that a variety of the down-regulated genes happen to be of M1 phenotype or associated with the M1 phenotype. so the pro-inflammatory phenotype, and many more are the upregulated that are associated with the M2 phenotype. So again, suggesting that this nanoparticle drug delivery system is biasing and promoting more of a pro-regenerative, pro-healing milieu to these based on macrophage uptake. And furthermore, we get a variety of upregulated gene pathways that are associated with healing with our nanoparticle delivered drug versus drug alone. And these are everything from extracellular matrix organization to blood vessel development and a bunch in between. So, so what I've been able to show you is that even though it was unexpected, our drug delivery system that targets bone is actually acting through macrophages and biasing the milieu into a much more regenerative phenotype. And that promotes fracture healing through that mechanism. And though unexpected, we were, frankly, kind of lucky, I would say, based on the choices that we made, assuming our initial model that we were impacting osteogenesis, that the drugs that we chose actually happened to have an impact on this M2 biasing as well. I often wonder if we had had negative results initially by choosing a different pathway, we certainly wouldn't have gotten here. And it's a really intriguing story. So hopefully convincing you that if you know more about your system and know that you're impacting, innate immune cells like macrophages, you can really leverage that. And that's exactly where we are. So now that we know that the macrophages really are the cell type that's the target that are scooping up these nanoparticles once they reach the bone fracture site, well, now we can have lots of good fun with that. So I'd like to really so I've highlighted a bunch of people here because both of these stories were really like long-standing efforts within my group, and so I have a variety of people that are just kind of pasted in addition to some more recent students. And so I'm really thankful for my awesome collaborators who are always asking the hard questions, and then of course funding. Yeah. Okay, I might have oversold the 5 minutes, but thanks for your attention. Nevertheless, thank you for being such a kind audience. I'm happy to have a lengthy discussion as long as you'd like, and really thankful to be here. So.
Thank you, Daniela, for the wonderful presentation. And in questions, yeah.
Hi, thank you very much for explaining your interesting project. I have a couple of questions. For the nanoparticles in both sRNA delivery and drug delivery, the major size was below or about 25 nanometer, and I imagine they clear out from the system after administration of drug or delivery of sRNA. So how long is the clearance, and especially for the aggregated nanoparticles?
Those are super good questions. Those are super hard questions to answer experimentally. So the aggregated particles, I don't even hasten a guess. So unfortunately, where we had again shown success, we don't see aggregation. But if one were to introduce aggregated nanoparticles, I mean, you could guess that it would be a very prolonged process because now you're left with, you need to hit the critical micelle concentration to disassemble into the dye blocks, but also need to degrade the glue, the protein glue. So it's actually really, I think I would envision that we could get some guesstimate based on some in vitro studies, but we haven't tried those. For the other systems, we don't see nanoparticles when we go back to do, for example, any of our measures. Now it's like a needle in a haystack, I'll give you that, because even the electron microscopy, when we're able to catch these and get some good images, you're just hoping that, and it's when you see them that you're like, oh, they're here, but you can't say anything about, beyond their morphology at that point. Certainly nothing about concentration. What we do see is that in a variety, in different tissues, the clearance is different, but we can see that the nanoparticles do have a resonance time. So from a fluorescence standpoint, we see drug there for 7, sometimes as long as 10 days, depending upon when we deliver it, and depending upon the tissue that we're delivering to. That doesn't mean that they're all gone, though. They just have reduced beyond the visualization, but it's a really good question, and we should follow up on it somehow. So if you have good ideas about how to really, rigorously evaluate that, especially when you have diminishing returns and diminishing concentrations, I'd be really happy to hear them.
Maybe on the chip as you're doing.
Yeah.
And the second question is about the dosage. When you, did you compare the dosage or the order of the dosage when you have it targeted directly or comparing to the traditional? So it's 10 times less, or how much is less?
So let me make sure I'm answering your question. So the dose of drug versus free drug?
When is traditionally administered, or when you target it, just administered?
Okay, so the doses of the untargeted and targeted are the same. But you're right in that, have we corrected for, we get a, 50% greater accumulation. We haven't done that. I would presume that if we were able to get the same concentration at the fracture site and avoid systemic toxicity, which is a challenge, although I will say we've done whopping doses of these nanoparticles and seen nothing aberrant happening, but then one would presume that we would get a similar downstream effect. It's a good question, though, asking, does the TBP have any impact on, healing? Yeah, good question.
Hi, Dr. Bennett. Good talk. I have 2 questions. One question is that you found the TBP AMPs is very good, except this peptide. Could we find other peptides which can also good for the AMPs? Have you tried?
Yeah, so we haven't. So from this original study, there actually were a couple other peptides that were identified. And I think that there are others that have been identified through this same mechanism of phage display for this and other systems and targeting. So absolutely, there's plenty of opportunity there. And now, I think that, one could question whether it's even necessary to do said studies, or you could probably narrow your studies a little bit using, modeling. There's such beautiful work in AlphaFold and otherwise where, you could do a lot of computational work to identify, how you might be able to develop new targeting approaches, or at least narrow the field a little bit. Yeah.
Yes, thank you. Another question is that when you investigate the mechanism for the TBP-AMP, it's targeted the M2 macrophages. You found some gene subregards, this M2 macrophage related gene and pathway subregard. But we curious is that M2 macrophage is always active the inflammation, and how it can treat to when the water heating happened is how can water heating.
Let me make sure that I'm answering the question that I think you're asking. So I think you're asking, we see this biasing towards more M2 macrophages, but they're going to be there anyway. Is that the question? So,
yeah, why not is the activate the M1 macrophage to cure to treat?
So, okay, let me again make sure I'm answering the right question. So, in the typical fracture healing cascade, you have M1s, well, just like any post-injury cascade of any immunity, you have macrophage that come in that are M1 typically, and they are the inflammatory cells that, recruit in other cell types and get everything primed and ready to go for regeneration. And then the M2s come a little bit later to coordinate the regenerative response. So I don't think I have a question there, but I think what you might be asking is like timing. Is timing important? Is that right? Yeah, timing is critical. So I guess that's the other thing. So I said something about the drug, but we also give this drug delivery system 3 days after fracture, and that's associated with how, like, when we can get the greatest accumulation and specific accumulation at fracture. But that's also exactly at the point at which you start to move from the inflammatory phase to the regenerative phase. So if we had started earlier or later, we might have seen a different result. So yeah. And I'm not saying that we're like, luck is always on our side. We've had lots of bad luck in our work, but yeah, I think that timing is key too. And we have looked at a variety of different timings. And this happens to be a really good point at which you can coordinate that transition and improve the M2 phenotype. And really the ratio of M2 to M1 is how we're reflecting it in a lot of our data. Yeah, very good question.
Thank you so much for the great talk. I have a question about M2 phenotype that you see in the bone regeneration, osteogenesis. Is it M2A or C? Because M2 has different subpopulation.
Yeah, so we have, absolutely, So we're so simplistic. So we're the classical engineer who's like, M1 or M2, we don't, I'd have to look really carefully again at the RNA-seq data. I suspect that, I'm not even gonna suspect, it's something that we recognize. And, I think the thing that we also need to be really concerned with is that having a long-term M2 macrophage in that area is not good. Like you'll end up with fibrosis, and we don't see that. So, it's a productive stimulus. But I can certainly dig in a little bit more. Yeah.
And do you see that the M2 macrophages is recruiting to that site, or it's M1 and M2 is recruiting to the site of the injury, or it's a monocyte that differentiating to M1, M2 from?
Also a really good question, and I don't have an answer to. So, yeah.
Yeah. Anybody else?
Do you have the online one?
Online one, couple of questions are there. Like the first one is, most of nanoparticles trigger innate immune responses in vivo? That's the first question. Second question is, are there any nanoparticle can specifically deliver the genes to the heart tissue?
Okay, so the first question was about innate immune system, like will all nanoparticles stimulate?
Yeah,
the innate immune response. Good question. I don't know that we have enough data to say that, but I think that there probably is. If you looked at the literature, I would say that in all likelihood, everything that gets internalized by cell, there's going to be some impact on innate signaling. I don't think that there's a way to circumvent that, but I think that through the careful consideration of the aggregated versus unimeric state, the types of proteins that are being dragged in with the nanoparticles, and the types of drugs, you can mute that response. Maybe not completely turn it off, but you can mute it. And then the second question, so can you deliver nanoparticles to, did I hear cartilage?
In the heart.
The heart, okay. Sorry, that was totally wrong. To the heart. I would say maybe. So it depends on which ones. And one would have to, develop. And I think that there is literature that suggests that there could be there are targeting mechanisms for the heart. So I would say yes, probably. But we can go back and use phage display to figure this out. And yeah.
Some other questions also. What is the byproduct of the nanoparticle after degradation? I would like to know about the degradation rate of nanoparticle. Have you looked at the protein absorption of nanoparticle, as well as especially when you attach it with the targeting ligand? It's a long question.
Yes, so many questions. So I kind of alluded to this already. So the nanoparticles inherently are like the diblock copolymers are not really degradable, I will say. But what does happen is that beyond the critical micelle concentration, they'll fall apart because they're not covalently bound to one another in those nanoparticle systems. So and those diblocks are of a molecular weight that they should get excreted. Have we carefully followed that? No. And should we? Yes. So that, and the degradation rate. So I think, like I talked through it a little bit with a past question. So we don't see nanoparticles at time points of healing. We don't see nanoparticle fluorescence beyond, a couple of weeks, a little bit less than that, usually 7 to 10 days after injection. And again, that doesn't quite get rigorously at that question, but it gives some inferences. Protein absorption after both in the presence and absence of targeting ligand. Yes, we've looked at this. Recollecting the data. So certainly with the zips, so I showed the zips, so I'm trying to suss out from the two different stories. So with the zips, yes, definitely we reduced protein absorption. I showed that. With the PSMA, so with the bone targeted drug delivery system, we have looked at protein absorption. I think it's pretty similar actually across the, like whether we have TBP or not. And I shouldn't know the exact data, but opening that file is slow right now. It's not too different. I don't know if it's statistically different, but it's not too different. The types of proteins could be different, and I think that would be interesting to look more carefully at. So doing proteomics, and we haven't done that super carefully.
Okay, thank you. Anybody else have a question? If not, I have more questions. When you synthesize this kind of different block of polymers, like for each batch, whenever you do the raft especially, so how do you control the molecular weight? How it is affecting the gene delivery efficiency?
Yeah, so we control the molecular weight and like the properties and the incorporation based on the reaction conditions. And so we spent a lot of time, making that really clean, I guess, and examining. And there are, best practices, I would say, that we've just learned from in terms of RAP polymerization and, the ratio chain transfer agent to initiator, for example, and the theoretical degree of polymerization, and there's a variety of, and it's dependent upon what your CTA is, what your initiator is, what type of initiation you're doing, solvent. There's a whole lot that goes into that. And then in terms of gene delivery efficiency, that was part of what we embarked upon in that first part of the study was, okay, once we have that all in check and very well controlled, can we then predict and alter gene delivery efficiency across different cell types and even different species. So I feel like we have a pretty good handle on that. That does not mean that we could then just pick up off the shelf nanoparticle and deliver it to cardiomyocytes and have it work really well without going back and looking at some of the optimization. Yeah.
Another question is one of the other polymer you have used styrene. The styrene, polystyrene plus malic anhydride, and you polymerize it. Styrene is so-called toxic in terms of, so how do you control the toxicity when you polymerize styrene?
Yeah, so it's interesting. Yeah, I know, really styrene. So when it's polystyrene, I think it's a lot more benign. And in our experience, we can jack up this. So maybe I shouldn't admit to this, But one of my students who started many, actually is Marion up in the left-hand corner. When she started in the group, we had, developed these nanoparticles and used them a little bit, but not a bunch. Well, I guess a little bit in vivo.
Yeah.
But I asked her, I was like, hey, can we just keep jacking up the concentration and see where we hit the toxicity limit? And we reached a solubility limit for injection before we hit a toxicity limit. So, and we were not just looking at can we kill mice? It was what's happening with liver, enzyme levels and kidney function. And we did a pretty careful evaluation. So we don't see that at least acutely. And we did look longer term. We looked out a month or so to the mice that received these doses. So in our hands, it hasn't been an issue. But, again, that's pretty superficial.
Yeah, okay. Thank you. And so thank you, everyone. Thank you, Dr. Benoit. And to give a big applause.